Welcome to RadOnc's Smart Review RadBio edition.
Great to be here.
Let's dive right in. Today, we're really zeroing in on clonogenic assays.
Yeah, these are, truly fundamental methods, aren't they, for understanding how radiation affects, well, a cell's ability to reproduce.
Exactly. And we'll look at both the lab-based stuff and the techniques for studying this in living organisms.
Absolutely. And just to frame this right, our main goal in RadOsec isn't just like zapping cells, it's stopping them from dividing, stopping that tumor growth.
Right, stopping that regrowth potential.
And these assays we're talking about today, they're how we actually measure that loss. That loss of reproductive ability.
Precisely.
Yeah.
So let's kick things off with maybe the most basic concept here, reproductive cell death.
Okay.
You hear about different kinds of cell damage from radiation apoptosis, necrosis, senescence.
Sure. Lots of different mechanisms.
But for us in radiation oncology, the thing that really, really matters is reproductive cell death. This is basically when a cell, it just loses that ability to keep dividing indefinitely, to make copies of itself over and over.
Like it runs out of steam, reproductively speaking.
Yeah, think of it like a photocopier that jams permanently. The power might still be on, the lights might blink.
But he can't make any more copies.
Exactly, it's reproductively dead. And this is why fractionation works, right? We damage cells, maybe don't kill them outright metabolically, but disrupt their division capability down the line.
That's a great way to put it. And it's so important to distinguish this from, say, metabolic death. A cell might look okay for a bit, might still be, churning away metabolically.
Right.
But if it can't divide to form a colony, for our purposes, it's a goner. That's the endpoint we need for tumor control.
Couldn't agree more. Which leads us straight into the second key idea, the in vitro clonogenic assay.
Ah, yes, the lab workhorse.
Often called the gold standard, isn't it?
Yeah.
For measuring cell survival after radiation, specifically in the lab, in vitro, in glass, basically.
And this goes way back. Puck and Marcus, 1956.
That's right, using HeLa cells, which is a story in itself.
Definitely. And the name clonogenic, it tells you what it's about. Can a single cell form a clone?
Can it generate a whole colony of identical daughter cells? And the principle, it sounds simple, but it's so powerful. The question is, can one single irradiated cell divide enough times, usually we say about 6 or 7 doublings,
Yeah, enough to be seen.
to form a visible colony? And the standard definition is usually 50 or more cells.
If it can do that after being irradiated, we count it as a survivor, reproductively speaking.
Okay, so that's the principle. Let's get down to the nitty-gritty. How do we actually do this assay, step by step?
All right, let's walk through it. Imagine you're in the lab coat. First up, you've got your cells. They're usually growing nicely in a flask stuck to the bottom in a single layer.
A monolayer, yeah, like little paving stones.
Exactly. Now, you need to work with individual cells, so you have to kind of pry them loose, break them apart.
That's where trypsin comes in.
Right, the enzyme trypsin. It's like a gentle chemical spatula, breaks the cell-to-cell and cell-to-dish connections. Now you've got a suspension of single cells. Your paving stones are now individual marbles floating around.
Okay, got the single cell suspension. What's next? This is critical.
Absolutely critical. Counting. You have to know exactly how many cells you're starting with.
Use a hemocytometer, maybe an automated counter.
Whatever you use, it needs to be accurate. It's like counting your seeds before you plant them. Fundamental for the math later.
Totally. So, counted the cells. Now what?
Now you plate them. You take a known number of these single cells and put them into fresh Petri dishes with growth medium, their food.
And here's a key detail, right? The number of cells you plate isn't the same for every dish.
No, definitely not. It depends entirely on the radiation dose you plan to give that dish. If you're hitting it with a high dose, you expect way fewer survivors.
So you need to plate way more cells initially to even get a countable number of colonies at the end.
Precisely. And the flip side is true too. For your control dish, 0 gray.
Always need a control.
Always. For that control, or very low-dose dishes, you actually need to plate fewer cells.
Why is that?
Because if you plate too many, and they all survive and grow, the colonies will just merge into one big blob. You won't be able to count individual ones.
Ah, right. You want distinct islands, not a continent.
Exactly.
Yes.
Nicely separated colonies are key for accurate counting.
Okay, so we've seeded the right number of cells in all our dishes, different numbers for different planned doses, plus the 0 gray control.
Now comes the zap. You take the dishes designated for radiation and expose them to your planned doses. One dish gets 2 gray, another gets 4, another 6, and so on.
And then Patience.
Lots of patience.
Incubation time. You stick them in a nice warm incubator for what? Usually 1 to 2 weeks?
Yeah, depends on the cell line. Some are faster, some slower. You need enough time for a single surviving cell to multiply enough times to form that visible colony we talked about.
Okay. Waited patiently. Hopefully we see some colonies forming. Now what? Now we need to actually see them clearly and count them. So you fix them first, basically preserve them, stop them growing.
And then stain them.
Usually with something like crystal violet. Turns the colonies this deep purple or blue color, makes them pop visually.
Like highlighting the winners. Easy to count under a scope, or even sometimes by eye if they're big enough.
Okay, done all that. Counted the purple dots on each dish. Are we done? Can we just say, okay, the 1,000 cells plated, 40 colonies survived at 6 gray, so survival is 4%.
Ah, tempting. But no, not quite that simple, unfortunately.
Why not?
Because you have to account for the fact that even in your control dish, the one that got no radiation, not every single cell you plated actually managed to form a colony.
Ah, right. There's some inherent inefficiency in the process, or maybe not all cells were perfectly healthy to begin with.
Exactly. And that inherent ability to form colonies under ideal zero radiation conditions. That's what we call plating efficiency, or PE.
Okay, PE, plating efficiency. So it's like the baseline success rate for these cells in this specific experiment.
Precisely. It tells you how good your cells are at forming colonies, even without any stress.
And how do we calculate that? We need the control dish again, right?
Yep, back to the zero gray control dish. The formula is straightforward. PE is just the number of colonies you counted on that control dish divided by the number of cells you initially seeded onto that same dish.
Colonies counted divided by cells seeded.
Right, and usually we express it as a percentage, so you multiply by 100%. PE, colonies counted, cells seeded, by 100%.
Okay, let's do a quick example. Say we seeded 100 cells in the control dish, waited a week, stained, counted, and we found 70 colonies.
We already know from our control dish that the plating efficiency was 70%, or 0.7.
70% plating efficiency.
Let's find the expected number of colonies if there was no radiation. We take the 2,000 cells seeded and multiply that by the plating efficiency of 0.7. Exactly. So in this particular experiment, even under perfect conditions, only 70% of the cells are capable of making a colony. That 0.7 or 70% is crucial. We need it to correct the results from the irradiated dishes.
Right, it normalizes everything.
Yeah.
Okay, so now we have our PV, our baseline. How do we get to the actual surviving fraction, the SF, for the irradiated cells?
Now we can calculate SF. The surviving fraction tells you the proportion of cells that kept their reproductive ability after radiation, adjusted for that PE we just calculated.
Makes sense. What's the first step?
First, for each irradiated dish, you need to figure out how many colonies you would have expected if there had been no radiation effect, just based on the PE.
How do we do that?
You take the number of cells you seeded on that specific irradiated dish and multiply it by the plating efficiency, but use the PE as a decimal. So if PE was 70%, you multiply by 0.7.
Okay, so expected colony cells seeded on irradiated dish multiple by PE as a decimal.
Got it. That gives you the number of colonies you'd predict just based on the plating conditions alone.
Then finally, the surviving fraction.
The surviving fraction, SF, is simply the actual number of colonies you counted on that irradiated dish,
The number we got after fixing and staining.
divided by the expected number of colonies you just calculated.
So SF, actual colonies counted on irradiated dish, expected colonies.
That's the formula. It gives you the fraction of cells that truly survive the radiation dose, corrected for the baseline plating ability.
Okay, let's nail this down with that detailed example again. Walk us through.
Sure. Let's say we plated 2,000 cells onto a dish.
Okay.
Then we irradiated it with, say, 8 gray, high dose.
8 gray. Got it.
We incubated, fixed, stained, and counted, and we found 32 colonies.
Only 32 surviving colonies from 2,000 cells plated after 8 gray.
Right. And let's stick with our previous plating efficiency, say it was 70%, or 0.7, determined from our control dish.
Okay. PE equals 0.7.
So, step 1, calculate the expected colonies on this dish without radiation. That's 2,000 cells seeded times our PE of 0.7.
2,000 times 0.7, that's 1,400. So we would have expected 1,400 colonies if there was no radiation effect?
Exactly. Now, step 2, calculate the surviving fraction, SF. We take the actual number of colonies we counted, which was 32.
And divide it by that expected number, 1,400.
Right. 32 divided by 1,400, plug that into a calculator. It comes out to about 0.0228. It's started to 0.023.
So SF, we 0.023.
Which means, after 8 grays, only about 2.3% of the cells retain their ability to form a colony once you've accounted for the plating efficiency.
That makes the radiation effect much clearer than just looking at 32 colonies from 2,000 cells. It's properly normalized.
Absolutely. And this SF value is what we plot if you do this for multiple doses and calculate the SF for each.
You plot SF, usually on a logarithmic scale on the y-axis,
Against the dose on a linear scale on the x-axis.
and bingo, you get your cell survival curve, that classic shape we all know.
That's it, the foundation of so much radiobiology, all derived from this in vitro clonogenic assay.
Incredible how powerful such a conceptually simple technique is. But, okay, we're in the lab, in a dish, it's clean, it's controlled, but it's not a living organism, is it?
Definitely not, and that's the limitation, right? Real biology is messy. You've got cell interactions, immune system factors, blood supply issues, hypoxia. None of that's really in the Petri dish.
Which is where the in vivo assays come in, studying this within a living system.
Exactly, in vivo. And people like Withers and his group did some really groundbreaking, clever work developing assays to quantify clonogenic survival right inside the animal.
Often focusing on stem cells in normal tissues because their survival is key for tissue recovery after radiation.
Let's talk about some of those key in vivo assays for normal tissues. One of the earliest, really historically important ones is the spleen colony assay. Till and McCulloch.
Right, focusing on blood-forming stem cells.
Hematopoietic stem cells, HSCs, in the bone marrow. The method is ingenious, really. You take bone marrow cells from donor mice, maybe irradiated, maybe not.
Okay.
Inject a known number of these cells into recipient mice. But here's the trick. The recipient mice have been given a lethal dose of radiation first.
To wipe out their own bone marrow, create space and empty niche.
Exactly. So the donor HSCs can potentially home to the bone marrow or the spleen. And if they survive and proliferate after about 9 or 10 days.
You look at the recipient mouse's spleen and you see these little bumps, these nodules.
Right. Visible colonies on the spleen surface. And the crucial finding was that each colony likely came from a single surviving hematopoietic stem cell from the donor.
Amazing. It was proof of concept for HSCs and let them measure their radiosensitivity in vivo. Foundational stuff for stem cell biology, too, not just radiobiology.
Absolutely. Okay, another major one. This one pops up on boards all the time. The jejunal encrypt assay. Withers and Elkind were key here.
Ah, yes, the gut. Jejunum, part of the small intestine. Very relevant for radiation side effects.
Definitely. Here, you're looking at the stem cells hiding down in the base of the crypts, those little pits in the intestinal lining.
What's the method?
You irradiate mice, then you wait specifically about 3 days.
Why 3 days?
That's typically when the damage is peaking, but also when any surviving stem cells are just starting to kick into gear and regenerate the crypt line.
Okay, 3 days post-radiation, then what?
Then, unfortunately for the mouse, you sacrifice it, take out the jejunum, section it, and look under the microscope.
And you're looking for
regenerating crypts. You count how many crypts look like they're regrowing, usually defined as having, say, 10 or more actively dividing cells.
And the big assumption here is
that each one of those regenerating crypts started from a single surviving crypt stem cell.
so by counting regenerating crypts per gut circumference, you get a measure of stem cell survival in this rapidly dividing tissue.
Exactly. A real workhorse assay for studying gut radiobiology, repair, sensitizers, protectors. Very important.
Okay, moving from the gut to the skin. There's a skin assay too, right?
Yep. The skin colony assay. Again, Withers was heavily involved. Target here, epidermal stem cells, the ones that renew our skin.
How does that one work? Seems tricky on skin.
It often involves irradiating a specific patch of mouse skin. Sometimes they do this clever thing where they shield a central spot, irradiate a ring around it, and then give the central test area the actual dose.
Why the shielding?
To make sure any regrowth you see in the test area really comes from stem cells within that area, not cells migrating in from the edges.
Okay.
Keeps it localized.
Makes sense. So irradiate the test patch, then wait.
Wait quite a while, weeks usually.
Yeah.
And you look for little visible bumps or nodules of regrown skin appearing within that irradiated area.
And each nodule is assumed to be a colony.
Originating from one surviving epidermal stem cell, so you just count the bumps. Measures skin stem cell survival in situ.
Clever. Okay, one more normal tissue assay. The testes.
Right, the Kestis stem cell assay. Again, Withers' lab contribution. Focusing on the spermatogonial stem cells, the precursors to sperm.
How's that done?
Irradiate the mouse testes. Then you wait even longer, maybe 6 weeks or so.
Why so long?
Need time for the more mature, differentiating sperm cells, which are also hit by radiation, to clear out. And for any surviving stem cells to repopulate the seminiferous tubules, the little tubes where sperm production happens.
So after 6 weeks, you examine?
Cross-sections of the testes under the microscope. You score how many tubules show signs of active spermatogenesis, indicating they were successfully repopulated by a surviving stem cell.
And these spermatogonial stem cells are quite radiosensitive, aren't they?
Very much so. So this assay is really useful for looking at radiation effects on male fertility and reproductive health.
Okay, so that gives us a good toolkit for normal tissues in vivo. What about tumors? How do we measure clonogenic survival of cancer cells in a living animal?
Great question. We need ways to assess tumor response too. One classic method is the TD50 assay. TD50 stands for tumor dose 50% take, sometimes called a dilution assay.
TD50. Okay, what's the principle?
It measures the tumorigenicity of a cell population. Basically, how many cells does it take to start a tumor? You take tumor cells from donor animals, maybe control, maybe irradiated.
Right.
You make serial dilution samples with fewer and fewer cells. Then you inject known numbers of cells from each dilution into groups of recipient mice, usually syngeneic mice.
Meaning genetically identical to avoid immune rejection confusing things.
Exactly. And you wait and see which mice develop tumors. The endpoint you're looking for is the TD50 value, the number of injected cells that resulted in tumors growing in 50% of the animals in that group.
Okay, so you get a TD50 number for control cells and a TD50 number for irradiated cells. How does that give you survival?
Well, if the radiation killed cells or stopped them from being able to form a tumor, you'll need more irradiated cells to achieve that 50% tumor take rate.
So a higher TD50 for the irradiated group means the radiation was effective.
Right. The surviving fraction here is calculated a bit differently. It's usually TD50, control, divided by TD50, irradiated. A higher TD50, irradiated, leads to a lower SF. It's measuring the loss of tumor-initiating ability in the population.
Got it. It's a population measure, not single-cell colonies like before. What else for
There's the lung colony assay developed by Hill. This one's interesting because it relates to metastatic potential.
How so?
You take your tumor cells, again from control or irradiated donors, but this time you inject them intravenously.
To the bloodstream.
Right. They circulate. And some will get trapped in the capillaries of the lungs. If those trapped cells are viable, clonogenic, and can actually grow in that new lung environment,
they'll form metastatic colonies in the lungs.
Exactly. After a few weeks, you examine the lungs and count the visible tumor colony. The number of lung colonies reflects the number of injected cells that were capable of surviving, lodging, and proliferating in the lung.
So it tests not just survival, but also aspects of the metastatic cascade. Useful. Any others?
One more key one. The in vivo, in vitro assay, the name kind of gives it away.
Hybrid approach.
Yeah, it combines the best of both worlds. You treat the tumor in vivo, inside the animal, maybe irradiated right there in the mouse.
So it experiences the real tumor microenvironment, hypoxia, everything.
Precisely. Then, after the treatment, you excise the tumor, break it down into single cells.
And plate those cells in vitro for a standard clonogenic assay, just like we discussed at the start.
Exactly. So you get the realistic in vivo treatment conditions combined with the precise quantitative readout of the in vitro colony counting. Very powerful technique.
Makes sense. And I guess you could do similar transplant things with normal cells too.
You could, yeah. People have done assays where they irradiate normal cells, like from mammary gland or thyroid. Make a suspension, transplant them into the right spot in a recipient animal, like a cleared fat pad for mammary cells.
And then score if they form functional structures like little glands or follicles.
Right. Measuring survival based on the ability to reform functional tissue units. It's another way to assess stemprogenitor cell survival in vivo.
So it's clear we have this amazing range of tools, from the petri dish to the whole animal, to study how radiation affects cell reproduction.
Absolutely. And these assays, both types, they're the engine that generates the cell survival curves. Those curves are fundamental.
They give us that visual, quantitative picture of dose response.
And they let us calculate those key radiobiological parameters, things like D0, which tells you about inherent sensitivity, or SF2, the surviving fraction at 2 gray.
SF2 is super relevant clinically, isn't it, given how common 2 gray fractions are?
Hugely relevant. And understanding these parameters helps bridge the gap between lab findings and clinical observations.
And thinking about research, these assays are indispensable, right? For comparing, well, everything.
Oh, totally.
Yeah.
Comparing different radiation types, Photons versus protons versus carbon ions, different fractionation schemes, hypofractionation, hyperfractionation, testing new drugs, radiosensitizers to boost tumor kill.
Or radioprotectors to shield normal tissues.
Exactly. And understanding why some tumors are just naturally more resistant than others or why some normal tissues are more sensitive. It all comes back to these kinds of measurements.
And even though we don't run a clonogenic assay on Mrs. Smith before her treatment.
No, definitely not clinically for individual patients.
The principles we've learned from decades of these assays, they completely underpin how we think about radiation therapy planning, dose prescription, balance and cure, and side effects.
Couldn't said it better. They form the biological rationale for much of what we do every day.
Okay, perfect time to maybe crystallize some of this into a few clinical pearls. Key takeaways for everyone listening.
Good idea. Pearl number 1. Remember what survival means in radiobiology. It's reproductive integrity.
The ability to make a colony, not just looking alive metabolically.
Right. If it can't divide long-term, it's functionally dead for tumor control.
Pearl number 2.
The in vitro clonogenic assay is a gold standard in the lab. You have to understand PE and SF calculations.
Plating efficiency and surviving fraction. Know how to calculate them, know what they mean. It's bread and butter stuff and likely on exam.
Definitely. And pearl number 3. In vivo assays are crucial because biology is complex.
The Petri dish isn't the whole story. You need the context of the living organism, microenvironment, immune factors, all of it.
Exactly. In vivo assays give you that more complete biologically relevant picture.
Excellent, pearls. Ready for a quick board bliss? Let's test ourselves.
Let's do it. Hit me.
Okay, question 1, revised carefully this time. An investigator plates 500 cells in a control dish and counts 150 colonies. In a parallel dish, 5,000 cells are plated, irradiated with 4 gray, and 60 colonies are counted. What is the surviving fraction, SF, at 4 gray? A, 0.012, B, 0.03, C, 0.04, or D, 0.12?
Okay, deep breath. The correct answer is C. Explanation. First, calculate the plating efficiency, PE. This is 150 divided by 500, which equals 0.3. Then multiply this by 100% to get 30%. Next, to find the surviving fraction, SF, we first find the expected number of colonies without radiation for the second dish. 5,000 cells seeded times the plating efficiency of 0.3 equals 1,500. Finally, the surviving fraction, SF, is the actual colonies counted, 60 divided by the expected colonies, 1,500. 60 divided by 1,500 equals 0.04.
Here. Okay, next one.
Question 2. Which in vivo assay specifically involves irradiating mice, then waiting 3 days before looking at intestinal cross-sections for regenerating crypts? A. Spleen colony assay. B. Lung colony assay. Skin colony assay. D. Skin colony assay. D. Jejunal crypt assay. The key here is intestinal crypts, and that 3 days time point. That screams D. Jejunal crypt assay.
Correct. D it is. Question 3. What is the primary outcome measured by the TD50 assay? A. Rad dose for 50% tumor cure. B. Rad dose causing 50% animal death. C. Time for 50% apoptosis. D. Number of tumor cells needed for 50% tumor take in recipient.
TD50, tumor dose 50% take. It's about the number of cells needed to get tumors in half the animal, so D.
Exactly right, D. Last one, question 4. Which technique involves treating a tumor in vivo but then assessing cell survival by plating those cells in vitro for colony formation? A. Standard clonogenic assay. B. Lung colony assay. C. TD50 dilution assay. D. In vitro assay.
Treatment in vivo, plating in vitro. That's the definition of D, the in vivo in vitro assay.
Fantastic. Nailed the blitz. So just to quickly wrap up what we covered in this deep dive.
We hit reproductive cell death, why it matters more than just metabolic status.
We went through the in vitro clonogenic assay step by step, especially hitting those PE and SF calculations hard. Remember the formulas?
Then we toured the in vivo world. Spleen colonies, jejunal crypts, remember 3 days. Skin colonies, testi stem cells for normal tissues.
And for tumors, TD50 for tumorigenicity, lung colonies for metastatic potential, and that hybrid in vivo in vitro approach.
And we tied it all back to how these assays generate survival curves, give us parameters like SF2, and form the scientific basis for a lot of clinical radiotherapy.
It's really foundational knowledge for anyone in the field.
Absolutely.
All right, that brings us to the end of this session. Remember, you can complete practice oral boards over at radonsmartlearn.com.
And definitely subscribe to Radon Smart Review so you don't miss our next deep dive.
Thanks so much for tuning in.